In this video, I want to walk you through how we can find a specific fragment out of a very large number of fragments, say from an entire genome, that has a particular sequence we're after. In the paper we're considering this week, the fragment we're after is the fragment that into which a transposon has jumped. So we'll start here with some genomic DNA. I'm just showing a short stretch of it here, and we're going to color it blue. And now a transposon is going to be introduced into the genome. It's going to hop, and when it hops, it obviously creates an insertion. So you can see it's moved into the right side of the genomic DNA. If we now choose to cut this DNA with a restriction enzyme that cuts but not inside the transposon, then we will generate fragments, only one of which will be the one that contains the transposon. But how can we find that fragment? So what we're going to do once we have the genomic DNA cut is we're going to run a gel. So this is an agarose gel. Agarose is related to agar, which we use for solidifying media in microbiology. And agar is just a polysaccharide that when you heat it up, it melts or dissolves into water, and then as it cools, it forms a matrix that holds that water in a jello-like consistency. And the more agarose we add, the tighter the pore size in that jello-like material. So we have our gel, and now we're going to load our cut DNA into it. So we're kind of showing them here as little pieces. And in this simple case, we're just going to have our two fragments here, the short one and the longer one that has a transposon in it. So now we're going to subject this gel. This gel is immersed in some buffer that will help carry electric current, and we're going to subject this gel to an electric field. Now, because DNA is negatively charged in an electric field, it will move toward the positive pole, and so it will start to move. If I can get my guy to move here, and as it moves, the fragments will separate, and the smaller piece will run faster than the larger fragment, and eventually get to its final destination when we stop the gel. Now, in order for us to detect the fragment of interest, we need our DNA not to be double-stranded, which it is now, but single-stranded, and we'll see why in just a second. To make the DNA inside the gel single-stranded, we're going to treat the gel in an alkaline buffer that will denature or melt the double strands into single strands. I've kind of shown them separating quite a bit, but that's just for sake of our vision here so we could see them. But we basically made the DNA single-stranded. The fragments are separated. We'd like to keep that separation, yet somehow get to them. So now what we're going to do is a technique that's been around now for close to over 50 years, known as a southern blot. And it's called a southern blot because it was invented by a researcher by the name of E.M. Southern. So we need a tray, indicated here by the little trapezoid at the bottom. We're going to fill that tray with some buffer. Into that tray, we're going to put a little stand that will allow us to rest our gel on it. We're going to put that stand into the buffer. We're going to then put a piece of filter paper on top of the stand. It will allow us to then soak up the buffer and bring it to the gel. We then put on our gel, and I've indicated here, even though we cannot see the DNA in the gel, the relative positions of the small piece and the larger piece that is a transposon. We make sure there are no bubbles in between the gel and the paper so that buffer can freely flow into the gel at all locations. We're next going to add a piece of special paper, indicated here at the bottom of the pile as sort of in gold, and that paper is going to trap DNA when the DNA hits it. And then we put on top of that lots of pieces of filter paper and paper towels that are basically going to soak up liquid, and on top we kind of hold that all together and maintain good contact with some sort of light weight that will hold everything down. Now what will happen is the buffer will move up from the tank, up the filter paper, and through the gel equally, and by capillary action, and will soak into the dry piece of the paper above. And this will actually carry the DNA along with it, but now at a right angle to the direction in which it traveled by electrophoresis. And here we see the DNA move up until it leaves the gel and gets trapped on the piece of paper that's laying on top. If we disassemble this apparatus and pull the piece of paper off, if we could actually see it at a molecular level, we would see that the DNA is now on the surface of the paper in the same pattern in which it was in the gel. But we cannot really see it yet. We need a way to go find it. And the way we're going to go find it is to use what's called a probe. Now I'll remind you that the DNA on the surface of this paper, which is now called a blot, is single-stranded. We're going to use as our probe a single-stranded DNA to find the sequence we're after. In this case, we want to find the fragment with the transposon in it, so we're going to use a small piece of the transposon, single-stranded, as our probe. That probe will move until it finds, and this is going to be immersed in liquid, it'll move around and bump into the blot at different points, but at some point it will actually hit its complementary sequence, and when it does, voila, it binds to it. We can follow it if it carries some sort of marker, such as radioactivity or some other fluorescent tag or something like that, and we can determine what fragment is actually the one we're after. So this is known as a southern blot. It is a tried and true technique that is used by researchers around the world, and again, it's based solely on the simple idea of two strands of DNA binding to each other via complementarity. I hope this video helps.